Now, I would like to move on to introduce our keynote speaker. We are thrilled to have, again, Dr. Patty Brennan from the National Library of Medicine offering us some remarks on context for this important work that we're all doing here with MCBK. Dr. Brennan is director of the National Library of Medicine at the National Institutes of Health. NLM is the world's largest biomedical library and producer of digital information resources used by scientists, health professionals, and members of the public. Since becoming director in August 2016, Dr. Brennan has positioned the library to be the epicenter of biomedical data science at NIH and across the biomedical research enterprise globally. Her leadership has led to the development of a new strategic plan that refocuses and enhances NLM's research, development, training, and information systems. By leveraging NLM's heavily used data and information resources and programs, Dr. Brennan is strengthening and advancing NLM's data infrastructure to accelerate data-driven discovery in health, engage new users in new ways, and develop the workforce for a data-driven future. Prior to joining NIH, Dr. Brennan was the Lillian Molman Bascom Professor in the School of Nursing and College of Engineering at the University of Wisconsin in Madison. She also led the Living Environments Lab, which is now known as the Virtual Environments Group, at the Wisconsin Institute for Discovery, which develops new methods for the effective visualization of high-dimensional data. Dr. Brennan is a pioneer in the development of innovative information systems and services, and her professional accomplishments reflect her background, which unites engineering, information technology, and clinical care to improve public health and ensure the best possible experience in patient care. Dr. Brennan holds an appointment as Associate Investigator in the National Institute of Nursing Research, Division of Intramural Research, where she directs the Advanced Visualization Laboratory. Dr. Brennan received a Master of Science in Nursing from University of Pennsylvania and a PhD in Industrial Engineering from University of Wisconsin-Madison. Following 7 years of clinical practice in critical care nursing and psych nursing, she held academic positions at Marquette University, Case Western Reserve University, and the University of Wisconsin-Madison. As a past president of the American Medical Informatics Association, Dr. Brennan was elected to the National Academy of Medicine in 2001. She is a fellow of the American Academy of Nursing, the American College of Medical Informatics, and the New York Academy of Medicine. In 2020, Dr. Brennan was inducted into the American Institute for Medical and Biological Engineering. And this College of Fellows is among the highest professional distinctions accorded to a medical and biological engineer. Dr. Brennan was honored for her outstanding contributions to biomedical informatics, leading to the acceleration of healthcare technologies to enhance patient self-management. What an impressive biography and list of accomplishments and wonderful perspective. Dr. Brennan, thank you so much for being here, and I'll turn the camera over to you.
Thank you very much, Rachel, and good morning to everyone. I want to begin by thanking Rachel and Chuck for their leadership in this really important initiative. Computable biomedical knowledge is absolutely critical because we know that health occurs where people are, not where professionals work, and we must get the knowledge to them wherever the professionals greet their patients. The National Library of Medicine is a partner with the MCBK activities for the last few years. We continue to want to support and see this area grow, but we're in the era of COVID-19. I'm speaking to you from the second floor of my mother's home in Bryn Mawr. I suspect all of you are in places you never thought you would be at this point in the summer. I'm here today to give you some introduction to how the National Library of Medicine is operating in the COVID-19 era, still going forward with our strategic plan. As you know, the National Library of Medicine developed a strategic plan about 3 years ago to accelerate discovery and data-powered health. We have 3 pillars of that plan: accelerating discovery and advancing health through data-driven research. And man, is this taking off like crazy. Reaching more people in more ways through enhanced dissemination and engagement. And again, who knew that the COVID-19 era would just grab every strength the NLM has to reach people where they are, and finally to prepare and to build a workforce. But today I'm going to be speaking to you specifically about 2 areas that the NIH is investing in: advances in literature distribution and advanced analytics. But first, I'd like to take a few minutes to tell you what we're doing in response to the COVID-19 challenges. The National Library of Medicine is committed to advancing science and clinical care and patient self-management. This is our banner ad on our homepage. Please note that, like all of NIH, we are coordinating our information flows through the CDC and through the NIH single portal. We are, in addition to this, operating under a maximum telework operation. So on the night of March the 13th, we were all told, go home and we'll let you know when to come back, and we're still not back. Our teams have been tremendous. They continue to operate. Our services have not missed a beat. We have continued to grow, which is essential because the healthcare challenges that we're facing under COVID-19 have demanded things that our system has never been able to produce, and we must stand there with them. The National Library of Medicine is pleased to have received $10 million in stimulus funds from the original Cures Act, and we've put that to good use already. We've supported better chemical editing for coronavirus-related drugs. We've enhanced our collection of materials that are available electronically, and we're also working to support access to high-quality literature when public health and public libraries are closed. We have improved our PubMed Central submission stream to facilitate AI and machine learning. We've developed a portal within PubMed for COVID-19 literature, curated specifically by Jean Liu's group in our NCBI. And this is now taking into consideration not only the topical information or nature of the virus or the interventions, but also things like geography, grant numbers, population addressing. So please take a look at our COVID-19 portal. And finally, we have been accelerating rapid sequence submission and access through GenBank and the Virus Hub, making sure, as you may have well known, that the work we began on January the 13th, exposing the first fully annotated version of the Wuhan virus, now continues to receive virus submissions every 3 minutes. We are also working upstream, trying to improve the quality of clinical data for research and care. And this is where we intersect a lot with the MCBK group. We're improving implementation guidelines, training for standardization, and addition of codes to support COVID-19 related laboratory tests within LOINC, and also to expand our value set authority, FHIR API, to enable the standardized sharing of COVID-19 terminology updates. We continue to accelerate also and invest in research, trying to address questions of deep phenotyping, of text mining, and real-time surveillance. We are committed to mining clinical data for deep phenotyping models that can be used to identify or predict the presence of COVID. And as you know, this is not a single disease. It appears to have many manifestations, and detecting some of its most unusual presentations, whether it be the coagulopathies that are occurring or the incredible cardiovascular and pulmonary challenges, requires a deep understanding of many, many features that might be subtle in the clinical record. In addition, we're investing in AI and machine learning, analytics, and visualization tools to enhance image and clinical data to support clinical decisions in real time, particularly looking at how we can build algorithms to differentiate community-acquired pneumonias from other pneumonias, given the enormous respiratory presentation of this terrible disease that we're facing. And finally, we're expanding our investment in public health surveillance using viral genomics, health data, and social media to identify spread. We've just launched a very exciting public-private partnership with some of our investigators at the University of Alabama to begin to detect wastewater, the presence of the virus in wastewater, and to determine if that virus actually remains its infectivity. These things are going on in our intramural and our extramural research program. These are specific investments that we've expanded because of COVID-19. Now let me tell you a little bit about what we're doing with the literature, motivated by, but not restricted to, the challenges of COVID-19 and the coronavirus. On June the 9th this year, the National Institutes of Health launched what we refer to as the NIH preprint pilot. We have been asked to respond to the change in scientific communication, increasing reliance on preprints, increasing use of manuscripts that are not fully vetted by journals and peer review, but still may be important to society. So we have developed a pilot project to add preprints of research that was supported by NIH to PubMed Central. These preprints will be discoverable through PubMed, and they will also be stored fully in PubMed Central. Our preprint pilot is not a plan to build a preprint server for NIH awardees. We are working in close partnership with well-known preprint archives such as bioRxives and medRxives. Our preprint server is not placing NLM as a comprehensive preprint discovery resource. We are right now specifically working in this pilot, starting off with the COVID-19 literature and only focusing on research that is submitted preprints that are submitted in support of NIH research. We also are not creating a new requirement for investigators. You may remember that 3 years ago, the NIH allowed the submission of preprints as a credible product of research, as an interim product of research in our annual reports and in our final reports. So we've already encouraged investigators to make use of this communication tool. Now what we're doing is making their preprints discoverable. Our pilot objectives include the following. First, we want to increase the discoverability of NIH research, of early NIH research. Second, we want to test the viability of making preprints available via PubMed Central. Third, we want to learn more about the impact of accelerated discovery and open sharing of research on scholarly communications. And finally, and importantly for the COVID-19 challenge, we want to meet the needs of the research community during the public health emergency. Our phase 1 of the preprint pilot is restricted only to research that is related to coronavirus and COVID-19, which is still a very large number of papers. If you look to the right-hand side of the screen, you see the banner designation that will appear on the top of the citation to demonstrate that this particular citation that an individual is looking at is a preprint that has not been peer reviewed. In order to have a preprint appear in the pilot, our investigators must have deposited their preprint in a recognized preprint server. For those of you who are interested in machine access to our research and our literature, remember that we are also able to designate by toggling a switch that a particular manuscript is a preprint, so it can be excluded or included in searches that are done. The NIH preprint pilot now is almost a month old. We are making about, right now we have about 1,000 preprints available. About 400 were available in the launch. But we found that in the very first week that these preprints were available, they were accessed over 20,000 times. So we see a need, a demand to access preprints, and we're really interested in hearing from the communities of how useful is this? To what extent does this help you find research articles in a more current and contemporary fashion? Or are you worried that this may have contaminated the brand, our very pure brand of PubMed and PubMed Central? Our preprint pilot will be monitored very closely for the next 6 months. We propose a gradual introduction of other NIH-funded research, so you should be seeing increased numbers of preprints discoverable through the PubMed search engine and the PubMed Central repository. Now let's talk about what's new with PubMed. In 2017, we launched something called Pubmed Labs, which was the beginning of a test of a brand new approach to PubMed. Pubmed Labs provided us with a way to rethink PubMed to address both the stability that we can and accessibility we can have through cloud architectures, and to make use of modern search techniques. You see on the screen in front of you on the left side, the interface, a web-based interface, and what's relevant to this community on the right-hand side was our initial tries at a mobile interface. The new PubMed has been officially launched as of May the 18th of this year. It's been launched to great fanfare, some dismay by some librarians, and lots of feedback, and we want you to continue to give us feedback. We are preserving access to the legacy PubMed at least until the end of September for several different reasons, including some concerns that during this COVID-19 crisis, to switch a search engine was getting very complicated for people. Our goal with PubMed is to make it a new modern hub with a fast and reliable, intuitive search engine that connects people to the world's leading sources of biomedical information. But what we know is there are 30 million citations in PubMed, and connecting any investigator to all 30 million is actually not useful. We learned over time that 80% of the searches, more than 20 search results occur, and most of the time, no one goes beyond the first page. So if the article you need is on the second or third or fifth page, you'll never find it. So our new approach to PubMed was to develop a way to organize, retrieve, and present search results, not by reverse chronological order, but by a relevance-based ranking system. This relevance-based ranking system draws on a number of factors, about actually over 800 different features brought together that look at things such as the citation itself, the text, the natural language structure of the abstract, but also how frequently this particular citation has already been pulled, in what context has this been pulled. You are still able to get your results in reverse chronological order if that's what you desire, but we believe that this new approach to PubMed is actually going to be providing you with more rapid access to the citations that you find most important. The goals of our new structure, which is to leverage cloud architecture to provide high quality search in a fast and reliable fashion, to create a modern experience, and yet to retain the good, positive response we've had from the community in the past. Now, we know in many ways, mobile approaches to knowledge management requires restructuring the display screens under a concept called responsive design. So what we find now is we have rebuilt the interface to PubMed for mobile devices that follows a responsive design process. It allows for direct login to My NCBI, you see in the upper right-hand corner, and easy access to advances, filters, and the timeline. It's possible to create site exports of citations and share citations, and also to get links to outside tools that allow an individual to better see data that may be associated with a particular article, share a particular citation to an individual. So we move the concept of the literature into the conversation of science. We have two separate websites that initially, this is what our legacy PubMed has transformed into. And now what we see in our new resource is we have built the same structures into our new web-based as well as our desktop as well as our mobile. If you look closely, what you'll see is the articles are offered in an option to be sorted by a particular structure. If you look in the left-hand side of the screen here, you see a histogram. This gives you a time series summary of the number of articles appearing by year. So you can hone into the point of your search that is most relevant to the individual. Now, we've worked hard to make the PubMed experience useful and pleasant for people. In the legacy PubMed, you see what was on the screen now. Lots of text and very small text for those of us over 65, too small to read sometimes. We've made the text larger in PubMed 2.0. We've also been able to pull out the citations and allow for them to be exported in a .rs mechanism. We have also allowed you to put filters in, for example, to only get full free text or restrict to associated data or only for randomized controlled trials. And finally, we've allowed for exports that allows you, if you are logged into your My NCBI, to send to your clipboard or have your citations mailed to you. I encourage you to give it a try to the PubMed and let us know what you think of it. If you look in the right-hand corner of any of our displays, you'll see a small green box. This is your feedback to us. Please tell us how it's going. We recognize PubMed is 25 years old and we've become quite familiar. It's part of the everyday fabric of science. And yet we know that for some individuals, changing the fabric of science is unnerving and off-putting. The two changes that I've described for you, the improvements in the use of preprints and the changes in PubMed, are occurring during this very time that we are involved in an enormous growth of science around the coronavirus and COVID-19. I want to take just a moment to tell you a little bit about some of the NIH initiatives that are relevant both to science and then specifically to this community, to advancing the analytics that help expose the knowledge underneath the data. The NIH is deeply involved in accelerating vaccines. We are part of the Operation Warp Speed, and we are working in a public-private partnership with the National Institutes of Health, several private foundations, the Gates Foundation, and the Foundation for the National Institutes of Health, and the pharma communities under an initiative referred to as ACTIVE. You can find information about the ACTIVE profile on our website at the NIH. But essentially, the vaccine acceleration process is happening because there's been an enormous partnership between the pharma companies, between the research institutes and the research funders. Two key things are happening to accelerate the vaccine trials. First of all, there's been strong agreement for the use of master protocols, which will allow for example, using the same comparison group multiple times. There will be shared data safety and monitoring protocols in place. There will be laboratory parameters will be harmonized across various studies, and the monitoring boards will be not only monitoring for the conduct of individual studies, but the concert of studies together. We know for a fact that there are several promising vectors for vaccines that appear to be likely to be trustable by the end of this calendar year. We also know that some of our pharma companies are already investing in building those vaccines in the hope that they will be ready for distribution in January. This is a financial risk on the part of the companies, frankly, but because of the nature of the national crises to get a vaccine out into the public as quickly as possible, we see an enormous and unusual level of collaboration. The NIH is also deeply involved in the therapeutics for the COVID-19 clinical manifestations. While the dexamethasone study at the top left of the screen was conducted in the UK, it occurred in partnership with researchers here in the US. And what was discovered in the dexamethasone trial is that a simple, well-known steroid actually decreased mortality in the very sickest of patients who have COVID-19 presentation. Recognizing the challenge of coagulopathy, the National Heart, Lung, and Blood Institute began an anticoagulation program in early June. Over this summer, in the next couple of days, next couple of weeks, we're going to be seeing clinical trials for immunotherapies, for outpatient monoclonal antibodies, and the beginning of a phase 2, phase 3 trials for antivirals. You may have noticed from the beginning that these particular approaches, particularly antivirals, were not thought to be as promising initially, but now we're seeing some support here. Advancing the understanding of therapeutics requires that we accelerate the use of artificial intelligence and new methodologies. So I want to close my remarks today by talking with you about a program referred to as ABLE, the Artificial Intelligence for Biomedical Ethics Excellence Program. The ABLE program began as an initiative supported by, driven by an advisory committee to the director of NIH. They argued that we needed to be generating new data sets to publish and create criteria for machine learning friendly data sets and make better use of industry standards such as model cards and data sheets. Also cautioning us that we had to develop strategies for consent and data access, to publish ethical principles for the use of AI and expand our training. This set of initiatives was accompanied by a cautionary note that said, please do not invest in developing generalized machine learning techniques. NIH should do its investments in health-related or bioscience-related machine learning techniques. In addition, we should be investing and are focusing on exploiting the use of new data sets rather than trying to fix old data set. We are in the process now of launching a new series of initiatives, and I'd like to summarize these for you. We will be developing generation of new data sets through a common fund program, but first we have to identify the criteria for them and then develop the tools to gain consent at the very beginning of research projects so that participants know that their data will be used for more general services. And we must, of course, publish the principles for good-based machine learning information and approaches. Now, our series of initiatives look as they do on the table in front of you. There are 5 initiatives to develop data design centers. We expect within fiscal 21 to be releasing through the NHGRI a call for proposals for data design centers that will create not data repositories, but design centers that will allow for establishing of the rubrics for identifying, evaluating, and disseminating access to the tools and the data necessary for accelerating discovery. There's a great deal of ethical, legal, and social issues involved here, including rules for data provenance, accessibility, privacy, and representation. The second initiative, which will be partnered between the National Library of Medicine and the National Institute for Biomedical Imaging and Biomedical Engineering, includes development of tools, software and firmware to accelerate AI readiness, tools that instrument that generate AI data sets, software that speeds up the annotation and metadata completion. And as you know, metadata is one of the key challenges of better use of AI. Data enhancement strategies are designed to take existing data sets with supplements to improve their usefulness, their curation, attach metadata, do harmonization to make these useful for AI and machine learning strategies. But we will also be investing in what we call developing gold data sets. That is, creating brand new data sets that can be specifically designed to be exploited through AI mechanisms. And finally, we have to advance some strategies for assessing the quality and use of existing data sets. Our timeline looks like this. From year one in fiscal 21 through fiscal 26, we anticipate investing about $130 million in this initiative. So you will see new opportunities for analytics, new opportunities for data sets, and new opportunities for tools. We hope each of these will actually bring encouragement to the community in front of us so that you can also participate in these activities and these resources. Our plan for the future will produce rubrics that allow for the evaluation of good data sets, tools that accelerate the creation and metadata identification of data sets, infrastructure to host and promote access to data, and a group of AI-ready data sets that will be useful for society. Our plan is to accomplish this ambitiously over the next 6 years. We hope you'll join us in this process. Let me close now and thank you for your attention. Remind you that we're improving the literature and investing more in analytics, both of which we hope will be well aligned with the goals of these particular initiatives that fall under accelerating mobile computing, computable biomedical knowledge. Thanks for your time, and I believe I've left enough time for questions.
Yeah. This is wonderful. Thank you so much for those great remarks. It's wonderful to hear all that the NLM is doing now in this time of global crisis. I want to encourage everybody to submit comments or questions through the Q&A chat pod, and those will get relayed to me, and I will relay those to Dr. Brennan. Meanwhile, I wanted to throw a question at you and get your comments regarding the preprint concept that you move forward and how that might apply to some of what we're seeing. People are developing algorithms and models and potentially decision support types of tools around COVID, and there's this urgency to get them out there. Has there been conversation about preprint or moving forward analytic models and tools in a quick way?
So thank you very much for the question, Rachel. And those of you who heard me speak last year heard me, and even a bit the year before, heard me calling for, we need to do something better to make models discoverable, to provide a way for storing information about the validity, about the strategies for their verification, and the trustability of the models, the provenance of the models. We have a number of partnerships envisioned to be able to do this. I had envisioned at some point we might be thinking about a Pubmed for models, a place where model declarations could occur, and we could actually have a storage of executable models. Over the last 3 years, as we've evaluated this, we've determined that really the best place for this is to create community-based hubs, community-based organizations to store and allow for access to models, to allow for the interchange and the interaction. Because although it's certainly feasible for an individual to contact an author of an article and ask for more details or get a data set associated with the articles, the conversation around literature tends to be more formal and more done through other literature. Whereas the conversation around models, how did you use this model? What was your original settings? What kind of parameters? What kind of tweaks did you have to make it work? Is often the result of informal interaction. So we see a shift in scientific communication, and rather than requiring that models and analytics and decision tools fit into the formal structure of literature communication, we believe it's important to be investing in building new sources and new repositories that will be more accessible to the community. We're looking to our national network, our network of the National Libraries of Medicine, which has 8,000 points across the country of academic health science centers, public libraries, and individual libraries. They also have coordinating centers, and there is a proposal. Our new solicitation has just hit out on the street in the last, maybe 3 weeks. And there is a proposal within that to create a data resource, a data center that will become a dissemination point for tools such as models, visualization packages, and analytics. I would be interested to hear from this community the extent to which the expression of an analytical tool or a decision logic through a preprint deposited in a preprint server might be a valid strategy for allowing that information about that particular resource to be communicated. The challenge that I think we're going to run into is, where do we store the executables that might need to be offered and reused by others? I leave that to this community to guide us on how we should make those investments, but I am very willing to take some chances and do some investigation in this area.
Wonderful, thank you. And I think we have, following you, a wonderful panel of experts representing the Library of the Future and sharing some comments on that, which will be wonderful. We do have a related question here from Jia Jing on how the quality could be controlled for preprints. I think this is the issues you were getting at, Patty. I'm not sure if the preprints go through a peer review process, and how does that fit within what NLM typically does in terms of curating knowledge?
First, I say hi, Jaji. Second, this is a really good question, and it's a question it's probably the second most common question I'm getting right now. The first one, of course, is, what did you do to my Pubmed? I loved it the way it used to be. But let's talk about a couple things. First of all, I will not. The NLM has made a good partnership with the literature, with publishers, to make sure that the literature is, and the peer review is, the highest standard of scientific communication in the world. Yet we have seen terrible failures of this, particularly recently in the COVID-19 era, where the rush to publication has left us with untrustable literature, even in some of our flagship journals. So we do have to be careful about contrasting what we think is quite pure in peer review, which may not always be as clear as we'd like it to be, with what appears to be unfettered, which is preprints. The NIH has put some quality controls around preprints. The following are required. First, the preprint must be deposited into a recognized preprint server. And we've had conversations with bioarchives and with medRxives about what does that require. It does require an individual be able to describe the provenance of the manuscript. It does require that an individual be open to the commentary and the peer review. And that is a different switch of quality management. Now, instead of a blinded peer review by a journal, we have a public, if you will, conversation in the marketplace about the particular idea and the approach. We've heard from some scientists that they actually like this approach, that it makes their ultimate archival papers much better. But we also know that not every paper that appears in a preprint server ultimately gets to an archival process. This takes us to the third quality strategy that we're putting in place, Jaji, and that is that we are going to be crosswalking, that is mapping between preprints and subsequent articles that grow from those preprints. And this represents a combinatorial problem, because a single preprint may spawn more than one archival article, and one archival article may be drawn from more than one preprint. So we're working to be sure that we follow the mapping so that an individual is able to get the most current, most up-to-date manuscript. We are pointing to preprints in a preprint server and archiving the preprints of NIH-funded research in PubMed Central. We are not archiving the conversation, that is, the quality comments made by others about a specific preprint. Those will reside only on the preprint server. And I want to close by talking to you about what is one of the most serious and complicated quality control measures we have that gets instituted after the fact, and that is retractions. The National Library of Medicine works in partnership with the publishers and the preprint servers to accept their descriptions of articles through an XML submission or the full text article in an XML fashion. We do not review articles. We review and certify journals as being part of the National Library of Medicine's repository strategy. When an article is deemed to be inappropriate needing to be retracted for whatever reason, whether it's because an error was made in a data analysis or a claim of falsification has been supported, the journals must make the choice of retraction. So the recent experience we had with Lancet and New England Journal, we needed to wait until Lancet and New England Journal retracted the articles before we were able to note the retraction in our citation database in PubMed. Now it's a little bit different with preprints. Preprints have been retracted, and preprint servers, bioarchives in particular, have removed preprints, and we will be following the same process. If a preprint is removed from the preprint archive, it will be so noted as retracted in our citation database. Thank you.
Wonderful, thank you. And I know we just have 1 minute left. We have a pile of questions, but so we'll just take one more if you don't mind. Neil Sarka asks, is there embedded in the discussion of biomedical AI excellence, that will undoubtedly lead to a new generation of digital health tools, those that are patient facing, perhaps? So perhaps you could talk a few moments about some of these analytic tools and computable tools in how you see directing those to consumers.
Sure. And Rachel, I'm very happy to have you scrape the questions from the chat, and I'll be glad to answer them as I can and get the answers back to you by tomorrow.
Wonderful.
So Neil, your question's great and it's near and dear to my heart. The vision of AI as a biomedical enabler is not restricted to professionals. It's also envisioned as individuals. We are actually leveraging something really exciting that the COVID era has brought on to us. If you're following the news, you may have seen that NIH has received almost $2 billion of new funds, and about $1 billion that is going into accelerating the tools for testing under a product we refer to as Rapid Access to Diagnosis, or the RADx projects. The RADx projects have 5 different levels of tool preparation, from things almost ready to go into market that just need a little bit of acceleration in a Shark Tank type of model, to high risk, potentially high valued new approaches to testing that haven't quite been thought of yet. And through each of these, we are learning about accelerating the transition from development of a analytic tool to its implementation in a either professional or commercial grade device or patient focusing device, to the points of how to collect the data, how to report the data in a standard fashion and store it safely, and then to large scale deployment. So while the ABLE program itself does not have a specific tech transfer, within the COVID analysis and the RADx programs that we're running on campus right now, we suddenly have a big shift to looking at distribution as not only through scientific articles, but through devices and tools. So I think we're going to be seeing this as a landmark shift in the way NIH is supporting the transmission of knowledge into practice. Thanks very much.
Wonderful. Thank you. Thank you so much for joining us today and for sharing your perspective and expertise. This has been a wonderful kickoff to our day.